Yeah, should start streaming. Let's check. All right, so streaming is starting. Sorry about that, everybody. This class we are going to have more than one instructor. Um, it's going to be me and David Ishii and perhaps some other people. I was trying to get David in so we could both be live at the same time, but that didn't seem to work out. So, yeah, we're going to try to get that set up next time so it works. But David, he's watching and he's in the chat. So if you have any questions, feel free to throw them out there. And hopefully, David will be able to answer your questions for you. Um, so this is for the Genetics 101 class. This class is going to be 8 weeks long, and the live stream is going to be every Wednesday at 4 p.m. California time. Um, there's going to be lots of hands-on stuff to this class, and the hands-on stuff is going to take a little bit of time because we're going to be doing lots of PCR. We're going to send stuff off for sequencing and a bunch of other stuff. So try to keep up as best you can, but if you can't, don't worry about it too much. Um, just follow along as you can. All the lectures will be recorded and they'll be put on the ODIN YouTube channel. So if you miss a lecture, just go to the ODIN YouTube channel and find the Genetics 101 class and the class you missed and rewatch that lecture. So this class, Genetics 101, is going to focus mostly on DNA. That's going to be our big thing. DNA and genetics, and how you can use the tools of molecular biology to analyze and understand DNA and genetics. So first, what we're going to do is, I don't know the level of everybody here, so we're going to try to start at, a basic level, but hopefully for those of you who already have some knowledge on this, it'll be a refresher or provide you with some new information. In the Google Classroom, I posted a link to the molecular biology of the cell book. I suggest you download that. It's a good thing to have on hand anyway, and read the first chapter if you haven't, and use it as a reference so you kind of get an understanding of DNA. All right. So DNA, that's what we're going to start with. So every living organism we know has DNA, right? I mean, there are viruses that sometimes don't have DNA. They run off of like single-stranded RNA or something. But every quote unquote living organism we know of has DNA, right? And this DNA is what stores the genetic information. But literally this kind of blew my mind when I first started thinking about it, but all DNA is just atoms and molecules, right? So like DNA is literally composed of a sugar, right? Like sugar, phosphate backbone, and a nucleotide base. But it's just a molecule. These molecules store information based on the order that they are in, right? So DNA stores information that is turned into RNA and then is turned into proteins. Those proteins are what keep things alive and are what make things do stuff. So like your muscles moving and everything like that, that's proteins that are encoded in your DNA. Now, all the DNA that is in genomes is stored as double-stranded DNA, right? So what we have is we have two strands of DNA. We have one strand that we call the top strand or forward strand. And then we have one strand that we call the bottom strand or reverse strand. Now these strands are complementary to each other, right? So certain DNA bases can only pair with other DNA bases. This because the arrangement of DNA and the hydrogen bonds that form between the bases, right? So A can usually only pair with T and C can usually only pair with G. Obviously, there's always extenuating circumstances with everything in biology, but generally, this is what's always going to happen. So if your top strand is ACT, then your bottom strand is going to be TGA, right? Complementary. Now, the complementariness of DNA allows it to be used as a template, right? So if you have two strands, what happens is you can separate the strands and rebuild a copy of the genome by only copying one strand at a time, right? So if we remove the top strand here and we try to copy it, what we can do is the cell or us, we can put in DNA bases that are complementary, right? So if we had GTAA, then CATT would be put in, and then GCCAGT, right? You should know that. You should know A pairs with T, G pairs with C. That's the most basic, primitive thing that you should know to understand how DNA is working. This information here is going to be really essential in this class. It's the foundation for PCR, it's the foundation for sequencing and so many things. So understand that DNA is composed of A, T, G, and C. It's got two strands that are complementary together. Now, DNA always moves in what they call the 5' to 3' direction, right? So when DNA is being synthesized or polymerized, it's always happening in the 5' to 3' direction. So we call the left-hand side 5' on the top strand. Now this is based on the arrangement of the backbone. It's a weird name, I know, but just understand 5' means the start of the top strand and 3' means the end of the top strand. On the bottom strand, 5' is opposite 3' and 3' is opposite 5', right? Because it's complementary, it's reverse. So 5' to 3', 5' to 3'. That's how it works. So when we're doing something like DNA sequencing or amplifying DNA using PCR, which is a technique we're going to use over and over and over again in this class, the DNA is going to be amplified from the 5' to 3' direction. All right? You got it? Sound good? Any questions? All right. So let's go down a little bit. So DNA, there are many different types of DNA, but DNA in cells is in what we call a genome. Now, all a genome is it's a combination of a number of different chromosomes, or it could be just a single chromosome that holds all the DNA for the cell. All right, we're almost there. Right here, right? So genomes can be all different sizes. Now, there's not necessarily a genome size doesn't necessarily relate to the complexity of an organism, but bacteria usually have smaller genomes than yeast and plants and humans, right? But humans don't have the biggest genomes, right? There are a lot of plants that have way bigger genomes than humans. But the size of the genome, when you're talking about bacteria and stuff like that, the size can, generally be it's going to be smaller than the human genome, right? But it's not strictly related to complexity. So don't think that. Don't think that like if a genome is smaller or bigger than the human genome, that organism is more complex or less complex than the human genome. Now, the human genome has 3.2 billion base pairs, right? 3.2 billion. That's a lot. A lot of DNA. Now, it's not just in one giant chromosome, right? Humans have 23 chromosomes and they have two pairs of those chromosomes. They call this a diploid genome, right? So the genome size is just based on, for a haploid genome, so that just means one copy of the genome in the cell, right? But if you've taken basic genetics classes, you may have heard about that. Like humans have different alleles for things, right? So, for instance, sickle cell anemia is a very harmful disease, especially when somebody is homozygous for it, when they have two copies of the sickle cell anemia gene, right? But when you only have one copy of the sickle cell anemia gene, it actually won't kill you, and even though it's still a disease and hurts you, but it also makes you resistant to malaria. So the number of people who are heterozygous with sickle cell anemia in parts of Africa is a lot higher than you'd see in other places in the world, right? Because people get malaria a lot there. It's evolution in action, live in humans, right? So one thing that you have to think about when you're thinking about genetics, when you're thinking about sequencing organisms or sequencing humans, it's not always going to be the case where the gene that you sequence is the only copy of the gene, right? So you have to remember that there's two copies of genes in organisms like humans, and in a lot of other organisms. Some plants have gone through uh, crazy genome multiplication events and have like 5 copies of their genome, right? So genetics is a little bit more complex than what we thought of just like sequencing genes, but that's okay because it's still manageable and we could still work with it and we could still do a lot of experiments in this class with it. PCR is a great technique and gel electrophoresis are great techniques to learn about these things. All right. So I'm back. All right. Any questions so far? Any questions? No? We good? Okay. So in DNA, right? We're talking about DNA. The cool thing about DNA is not just something that you can read from. You can also turn RNA into DNA and read that, right? So a lot of our experiments and a lot of our science is based around DNA. Molecular biology is based a lot around DNA. This is not to say that you can't do stuff with RNA or proteins or these other things, but a lot of our technologies are based around DNA. So, like, if you want to sequence something, you're going to generally sequence DNA. It's more stable, it's easier to work with, but there are certain techniques, and what you can do is you can take the RNA and turn it into DNA using things like reverse transcriptases, and then you could use that DNA for downstream application. So this is what scientists used to do a lot back in the day. What they would do is they would amplify copies of genes from RNA, right? Turn them into DNA, and then put that DNA into a plasmid or something, or sequence it in order to understand what those genes were doing, right? Because the DNA is not just composed of regions that make proteins, it's also composed of all these non-coding regions and all this other stuff. So maybe you want to sequence just the important parts of a gene. And by important parts, I just mean protein coding regions. Obviously, other parts of the gene can be important, but you want to sequence just the protein coding regions. Then you would try to amplify the RNA that came from the gene, because RNA has all the introns and other non-coding regions removed. So you'd be able to look at just the gene that has the protein coding regions, right? Otherwise, the difference between DNA and RNA is just that thymine is replaced with uracil. Well, that's not the only difference. Also, the backbone is different. But when you're talking about the DNA bases, you have the 4 DNA bases. Thymine is replaced with uracil. What, did we miss it? I'm going back to it. All right. Anyway, so for the first week, the things that we're going to do is we're going to start by running an agarose gel. We're going to separate DNA on an agarose gel, and we're going to see how we can visualize DNA using molecular biology. Now, either you received a gel electrophoresis kit from us with a gel box and a power supply, or you have your own. So you should have received reagents from us that include a DNA ladder. And what you're going to want to do is you're going to want to run the DNA ladder out on the gel. So let me explain to you a bit about gel electrophoresis, right? So gel electrophoresis is how we analyze DNA on a cheap, inexpensive, and basic level. So gel electrophoresis, all it is, you have to think about it like a sieve, you know? Like a sieve that you're using, or something for cooking. So we have a matrix, which is made of agarose, right? So here's our gel. Generally, gels are in squares, right? And we have a well that's made with our comb. And DNA is going to run down this gel, right? So we have a strand of DNA, double-stranded DNA, right? And it's trying to move through a gel. Now, the gel is not 100% solid. It has different holes in it that allow the DNA to migrate. So what happens is that smaller DNA will move faster, right? Because it can go through the holes and make it through, while if a DNA is much bigger, it's going to move much slower because it's going to have a harder time trying to find its way through, and where to go. So agarose gel electrophoresis, what it does is it uses an applied voltage field, right? And this voltage, what it does is it carries the DNA through the agarose gel, right? Now, what you're going to do is the DNA is going to move in a specific direction. It's going to move from the black negative pole to the red positive pole. So when you put your gel in the gel electrophoresis box, you want to put the top of the gel where you load your DNA in the gel, near the black, and you want to put the bottom of the gel where you expect your DNA to travel near the red, right? Black to red, or else your gel's dead. Think about it like that. So what we're going to do in this first class, one of the first experiments, is we're going to make a gel and run out a DNA ladder. Now, a DNA ladder is just a bunch of chopped up pieces of DNA. So this is our well, and we're running out a DNA ladder. So what you're going to have is, as the DNA migrates down the gel, you're going to start to get separation between the DNA, right? So these are a bunch of little pieces of DNA. And this DNA is made at specific sizes. So somebody went in and amplified different sizes of DNA that they sell so we could use it as size markers. So for instance, if this is like a 100 base pair ladder, the first band is generally going to be something like 100 base pairs, then 200 base pairs, then 300 base pairs, right? So if we run our DNA on the gel next to it, our DNA is eventually going to stop someplace. Let's say it stops right here in the gel. And then we know our DNA is somewhere between 400 base pairs and 500 base pairs, right? Now, it's not going to give you an exact reading on how big your DNA is, but it's close enough. Now, the secret of the agarose gel is that the bands become further apart and the longer you run it. But you can actually see it even if you only run it a short period of time. Now, generally, you want to run an agarose gel at somewhere between 100 volts to 200 volts. Now, 200 volts might be a little too much. What happens at 200 volts is the solution starts to heat up. Now, this is because the resistance gets higher because the electrolytes in the buffer start to get electrolyzed, and then the resistance goes higher, so that causes heat to be generated as the current tries to run through the gel electrophoresis box, right? So now now the amps is also important. The current that's going through. You generally want to keep that between 100 milliamps and 300 milliamps. But generally, you don't need to worry about the amperage. As long as you keep the voltage at a specific level and make sure you have fresh buffer, you probably shouldn't heat up your gel too much, and it should be pretty good. Now, the more you heat up your gel can actually melt, and also makes your DNA run weird, right? Because your gel is starting to become depolymerized from heating it up so much, right? It's starting to melt. So your DNA can get smeared or run strange or all these other things. Now, how do we visualize the DNA actually in the gel, right? So we're putting DNA in here, we're running it through the gel, but how do we see the DNA visually with our eyes? Like, how do we see this? Now, generally, scientists use a couple types of molecules. The old thing that people used to use is ethidium bromide, ETBr. Now, ethidium bromide used to be really popular But people started to get scared of it because there were suggestions that it was toxic to human beings. Why is ethidium bromide toxic to human beings? Well, nobody really knows how toxic it is, or if it's really toxic, or if it's any more or less toxic than all the other things out there. Because the thing that makes uh, these dyes work is that they stick to DNA, right? So they bind to the DNA bases so that you can visualize the DNA. So when you start running your DNA through the gel, it starts to collect the DNA visualization dye, gel stain, whatever you want to call it. And as it collects it more and more, it gets brighter. Smaller bands aren't as bright because they can collect less gel, visualization dye. So sometimes when you're working with small pieces of DNA that maybe be like 100 base pairs or less, you should add more visualization dye, right? And when you're working with big pieces, you generally can add less. Now, there are multiple other different types of stains. We just use a generic gel stain that is supposedly much safer than ethidium bromide. But again, the studies aren't really, there's not really many studies to show that it's dangerous or not. But generally, it's good practice that when you're using gel stain in any way, that you're wearing gloves. Um, it doesn't hurt, but there have been no known cases of people getting cancer or anything from handling ethidium bromide or gel stain. But be safe. Wear gloves. It doesn't hurt. So now the way that the gel stain works is it's fluorescent, right? And the way fluorescence works is you shine a light on something. That's a terrible light bulb. Don't, I'm just learning how to draw with this thing. Light bulb. You shine light on the piece of DNA. The dye gets excited, and the dye emits light in a different wavelength, right? So the gel stain dyes that everybody uses generally absorb in the UV or blue range, and they emit uh, a, I think they emit what they emit above that. So what we do is we use a blue light to shine on the gel to visualize, and then we use an orange filter. Now, orange filters usually have a cutoff at something like 450 nanometers, which is around where the blue light spectrum is, right? So what we do is we block all the excitation light, and all we get to see is the emission light. So then what you see what'll look like little green bands on the gel. If you're having trouble seeing it, number one, you need to make sure that you added the gel stain to your gel. Number two, you need to make sure you're using the orange piece of film or orange glasses or whatever you have, holding that in front of your eyes. Then you need to make sure that you're shining your blue light or flashlight or whatever it is below this orange piece of film, right? If you're shining it above, it's going to block out all the excitation wavelengths and won't be able to get excited. So hold it below. Generally, what's good to do is to take a piece of the orange film, tape it over the camera on your cell phone, right? And then hold up your cell phone and shine the blue light. Then you could visualize, zoom in, and take a picture all at the same time. It works really great. And it's good to take pictures of every gel you run no matter what. It helps to understand what's going on and helps us to help you troubleshoot what's going on. Now, the final thing we need to learn about gel electrophoresis is that in order for the current to be carried across the gel electrophoresis box and through the gel, you need to use buffers, right? You need something with electrolytes in it that can be electrolyzed, that can carry um, the current through it. Now, so if you add tap water or distilled water, nothing will happen. Now, you could add stuff as simple as like sodium chloride, but generally that won't give you a good current or a good resolution on gel. So what we do is we use specific buffer formulations that help do the best for the gel and for the DNA. Now the most popular one is TAE, and that stands for tris acetate EDTA. And basically what this buffer does is it provides electrolytes and it provides EDTA so that the DNA, if there's any nucleases or anything around like that, the EDTA can protect it, and the tris acetate provides a buffered electrolytes that allows the current to run through the gel, right? Now, there are other buffers that people use, a lot of other buffers. Some people like to get pretty anal and stuff about their buffers. Some buffers give you better separation and visualization with smaller DNA. Some buffers give you better separation and visualization with larger DNA. But generally, if you use TAE, your buffer is going to be okay. It's going to do fine with no matter what DNA you run, right? So for this class, we use TAE, and probably for your life, you're probably going to use mostly TAE. So, stick with it. So, all right. Now, what else was I going to say about gels? There's one more thing that I wanted to tell you. There's a few things. So, one, gels can generally be reused, right? So, after you're done running your gel, you can break it up and then you can reheat it, remelt it down, and use it again to make another gel, right? Because you can imagine when you reheat it and remelt it down, all the DNA gets dispersed all throughout the gel. This is only in the case where you're not reusing the DNA in the gel. So sometimes scientists, what they'll do is they'll run the DNA out in the gel, they'll cut out the DNA band, and then they'll purify it from the gel. If you're doing that, you can't use a reused gel. But in general, you can reuse your gel. Each time you reuse it, I would add, 2 to 3 microliters more of gel stain, because some of the gel stain just breaks down or whatever. But you can generally reuse a gel, 3 to 4 times. I would try to keep it below 4 because gels also have TAE in them, right? You don't make a gel with agarose and water, you make it with agarose and TAE. That's a good indication whether if your gel is running funky. If you don't see bands of DNA like this and you just see kind of like a smear or some other wonky looking thing, try to remember if you use TAE or water. Generally, that's one of the first issues you can run into, right? But you can reuse your gel. It helps save you materials, helps save you money, even helps save you a little bit of time. All right. I'm back. Okay. So I see David's been answering your questions. Does anybody have any questions specifically about gel electrophoresis? Maybe? No? Hmm. So for your first experiment, you're going to run a gel, run out the ladder, take a good picture, right? Post it in the Google Classroom. Make sure you get it right before you proceed because once we start doing PCR, then you're going to have to make sure that, yeah, like David said, pro tip, don't touch the electrodes or don't stick 2 fingers in the gel box in the liquid while it's running. You'll get a shock. 1 finger's okay. 2, then it creates a circuit. Any questions? Any questions about gel electrophoresis? Anything? No? Any questions about what we talked about today? About DNA? Make sure if you have a chance, you read that chapter 1 in molecular biology of the cell. Um, Probably what will replace gels is capillary electrophoresis, if that's what you're asking. I don't know if what will replace GE in the future, if GE is gel or GE is gel electrophoresis. Capillary electrophoresis is probably the thing. It's just there's no cheap, inexpensive systems, but all it does the same thing as gel electrophoresis, but it's kind of digital. So it would just be like a computer telling you where each piece of DNA, um, when it came off, right? So it'd basically just be like a timer that's be like, all right, I had a band come off at this time point, at this time point, at this time point. So it's basically just like a time referential frame for gels. Um, we'll see. Somebody needs to do that. I mean, the problem with gel electrophoresis is it's so cheap and easy. It's really hard to get rid of, right? Because it's just so cheap and easy, right? You could probably run a gel with like Jello or like anything. How do you place the DNA in the gel electrophoresis? Not a dumb question, Chuck. So when you're actually creating the gel mold, if you look in our classroom, we should have we should have instructions on how to make a gel and how to load a gel. Um, what you're going to do is you're going to carefully pipette it into the different wells of the gel. So basically, you're just kind of like dropping it in these holes that you make in the gel mold. So you should have a gel comb. You should have received or will receive a gel comb and a gel tray. And the gel comb is basically just a bunch of little teeth like this. So then when you solidify the agar around it and you pull the comb out, you're left with a couple holes, right? And then you could just add the DNA to those holes. You mix the DNA with dye. I think in this class, some people will have gotten some master mixes that have pre-mixed dye. The DNA ladder should already have dye mixed in it, so you don't have to add dye to the DNA ladder. Um, So basically, you just add the DNA ladder directly to these holes in the gel, and it will it'll stay in there. So the DNA loading dye, usually that you mix with samples, sometimes depending, is composed of glycerol or a sugar. And what this does is it just mixes with the sample. It becomes heavier than water, so it'll stay inside these wells. You'll see. We have instructions. If you go to the classroom and go to the gel electrophoresis and analyze gel electrophoresis, Google to document tutorials, they should have a great description of how to do all this stuff, right? With pictures and everything. And if you have any questions about any of this stuff, please post it to the Google Classroom. But we have pictures and tutorials, and I think we even have videos. I don't know if the videos have been posted yet. What I'll try to do is get the video up of how to make and run gels and all this stuff. So it shouldn't be hopefully a problem. Just check out those tutorials and videos, and let us know if you have any questions. If you have any questions, feel free. Yeah, there are videos on there are plenty of videos on our YouTube and other people's YouTube. Feel free to find us on Facebook or the Google Classroom or email and ask questions, right? There's no stupid questions. Just ask questions. That's what we're here for. We're here to help teach you and here to help learn. In the beginning, we're going to try to do a lot of theoretical stuff, and we have a lot of documents and images and pictures and videos for all the hands-on stuff. But feel free to ask questions about it. All right? Any other questions? Anything else? So gels are for, you can quantify DNA using gels. Usually DNA ladders have standardized amounts. Gels are for finding length. So if, for instance, you're wondering if you have the correct DNA, you can do a restriction digest or something and use the length of the DNA to confirm that you do, the length of DNA in the restriction digest to confirm you do have the correct DNA. But DNA is all just about size and amount of DNA. That's what you can tell, and there's a lot of different things you can do from knowing the size and the quantity of DNA. All right. If there are no other questions, like I said, get started on the first 2 tutorials. Make a gel, load up some DNA ladder, and run it out on the gel, and take a picture and show us your results. That's your first experiment. All right. If nobody has any more questions about this stuff, then we will see you all next week at the same time, 4 p.m. All right. Thanks, everybody, for coming. Talk to you soon.